This is Graham Dick. I lead commercialization at Qualyst Transporter Solutions. We at Qualyst provide consulting and research services and products to help our clients implement in vitro studies. We are pleased that you have joined our webinar titled Hepatic Transporters and Clearance. It is my privilege to introduce our speakers. Dr. Ayman El-Kattan is Associate Research Fellow at the Pharmacokinetics, Dynamics, and Metabolism Department at Pfizer's labs in Cambridge. His research is focused on understanding the role of transporters in influencing drug disposition and oral absorption. He has investigated the utility of physiological-based pharmacokinetic modeling tools in projecting drug disposition and drug-drug interaction liabilities in man for new molecular entities. In addition, Ayman is a professor at the College of Pharmacy at the University of Rhode Island and serves on the executive committee of the AAPS Drug Transporter Focus Group. Dr. Kim Brouwer is Associate Dean for Research and Graduate Education at the University of North Carolina's School of Pharmacy and Keenan Distinguished Professor in the School of Pharmacy and Curriculum in Toxicology at UNC. She founded the UNC Pharmacokinetics and Pharmacodynamics Fellowship Program, and she directs an NIH-funded research program focused on hepatobiliary drug disposition, hepatic transport proteins, and development and refinement of in vitro models to predict in vivo hepatic drug disposition, drug interactions, and hepatotoxicity. Our speakers have each prepared remarks that will take a total of about 45 minutes. We want to leave time for questions. Please type your questions into the Q&A box. We will see the questions and ask Kim or Ayman to respond. At this time, I'd like to turn the floor over to Ayman.
Good morning, and thank you, Graham, for this nice introduction. I would like to appreciate Qualyst's commitment to educate the general public on the utility of transporters and how we can put these things in perspective. The title of my presentation is Application of Sandwich Culture Human Hepatocytes in Drug Discovery and Development. I will first have the outline. I will start with why we are interested in sandwich culture human hepatocytes. I will basically give an overview on the drivers for the interest in this technology. After that, I'm going to provide an overview of how we predicted clearance, drug interactions, and pharmacogenomics of new molecular entities for these compounds that are eliminated by active uptake by OATP. And this is going to be by providing an overview of Pfizer in-house model, and then discussing how we have used SEMSEP in combination with sandwich culture to enable that task. After that, I'm going to provide an overview and also a call of how we can actually invigorate the utility of human-based in vitro reagents to predict human clearance, drug interactions, and pharmacogenomics. And finally, I'm going to end up with the concluding remarks. Now, why we're interested in sandwich culture and why now? When we look at the past, let's say in 2004, there were a group of scientists in Pfizer that looked at the route of elimination of the top 200 molecules that were prescribed in the United States in 2002. What was interesting with that scholarship is that around 72% of these molecules, they were eliminated predominantly by metabolism, and to a lesser extent by renal, followed by biliary elimination. The fact that we are hearing that these are the top 200 prescribed molecules, this is actually an indication that these molecules were actually discovered and identified into the area in the time of around the 80s and early 1990s. So basically, it's a reflection of an era. And that era, like this is basically, was based on looking for compounds that are lipophilic in nature. And because of that, predicting compounds clearance was easy done by the use of microsomes and hepatocytes. Since most of the compounds that were metabolized, they were eliminated by CYP elimination. In addition, for the predictions of renal elimination, that was easily captured by things like allometric scalings. Now, this way of thinking was appropriate for that time, the area of the 80s and the 90s. However, there are major drivers of change that we are encountering as a drug industry. And that's actually shown with this slide. Sherry Koo from Legacy Wyeth led a team from 8 Pharma. Basically, the team task was to look at the changes in the drug disposition of the compounds that went into first in human and that were in phase 2. Basically, what the team did, they collected all the information about the disposition of 108 molecules. The outcome of that scholarship was really eye-opening. For example, we have seen a significant reduction in the contribution of metabolism to the overall disposition of the molecules. It went down from 70 down to 40%. We are starting to see other routes of eliminations that are actually contributing and driving the elimination of our molecules. This actually is a very major outcome that we need to keep in mind. In addition to all of that, when you think about our understanding of the clearance, we're actually observing that there is a major change in how we are approaching clearance. I remember in the early 90s when I started learning about the clearance concept, our scientists as well as faculties, they taught us that actually clearance is an additive process. When I say clearance, in this case, I mean specifically hepatic clearance. It is an additive process. It is the sum of the metabolic clearance, uptake clearance, as well as biliary clearance. The sum of all of that is going to give you the hepatic clearance. Now we know that this is not true. Clearance is not an additive process. As a matter of fact, it is an interplay of all of these processes. And to elaborate more on this thinking, we need to provide an overview of the extended clearance concept. In this diagram, as you can see, this is basically the passive intrinsic clearance, the uptake intrinsic clearance, and the metabolic and biliary elimination. Now, the interplay of all of these inputs will determine your hepatic intrinsic clearance. When you look at this equation, you may find it a little bit complex. However, if we take and make certain assumptions, our understanding of this equation that was established by Sandy Beng and later on invigorated by Yushi Sugiyama from Japan, it's going to be easier to comprehend. Now let's walk you through it. If you actually take a look at the metabolic and biliary clearance, and you make an assumption that it is significantly lower than the passive clearance of the molecule, combined with no active hepatic uptake, this equation is going to end up with this simple concept, where intrinsic clearance is basically equal to the metabolic and biliary elimination. There is another case where passive is actually extremely low. As a matter of fact, it's significantly lower than metabolic and biliary, and it is combined by significant active hepatic uptake that's mediated by OATP. If these assumptions are going to be established, you will find out that actually this equation is going to end up with this simple concept where intrinsic clearance is actually driven by a rate determining step, which is active hepatic uptake. In this case, even if your compound is going to be metabolized, metabolism as a rate is not going to have a significant impact on the systemic exposure of the compound. The last case is where all of these terms, all of these inputs that we just mentioned are appreciable. And this is where you're going to have the extended clearance, where we're going to have interplay of all of these concepts. Case 2 and case 3 is where you will have active hepatic uptake that you are going to be mindful of it to put things in perspective. Now, one of the things that we have recently done in Pfizer and we have published is the extended clearance classification system. This system was established mainly to enable us to identify the rate determining steps of our molecules early on in drug discovery. And this, we believe that if we're going to follow this approach, this approach will provide us with the guidance that's going to enable us to basically identify rate-determining steps, and based on that, we're going to be streamlining our investment to fit that thinking. The beauty about this approach is that it is based on 3 pillars, if you will, that are readily available in drug discovery. And these pillars are permeability, molecular weight, and ionization. Based on these 3 pillars, we are able to basically classify the compounds into 6 classes. Each one is going to be driven by a rate-determining step that is going to be different from the other. In this case, and based on the extended clearance concept, compounds that are acids and water ions and have a molecular weight higher than 400, they have a tendency of having hepatic uptake as a rate-determining step. Now, what we have observed that in addition to all of these things, which is the first one is the changes in the chemical space that's ending up with the changes in disposition, changing in our understanding for clearance and how to calculate it and put it in perspective. You are seeing a lot of pharmaceutical companies, many of them, especially in Cambridge as well as San Francisco, that they are actually very interested in the concept of hepatoselectivity. So why don't we define this concept? It's basically the achievement of pharmacological selectivity in the liver relative to other body organs as well as systemic exposure. Typically, this is defined by compounds that will have high KPUU, which stands for high free liver to plasma ratio. Now, why we in the industry is interested in this concept, the hepatoselectivity? It is because there are major diseases that are devastating a lot of our populations, and we need to find an effective cure to solve them. And these diseases are hepatitis C, NASH, liver cancer, not to mention hyperlipidemia. Our scientists in the pharmaceutical industries are actually trying to establish the hepatoselectivity based on using OATP transporters to put these things in perspective. And through the use of OATP 1B1 and 1B3, this is going to enable us to enable the hepatoselectivity, and as a result, improvement in the efficacy of our compounds, as well as a reduction and a significant reduction in the side effects associated with systemic exposure. Now, based on all of these things, I think you may agree with us that we need to change. This approach of one size fits all is no longer an option. To keep putting all the investment in human liver microsomes and hepatocytes as a driver for us to put clearance in perspective should change, and it should change now. Now, because of this, what you have observed is that colleagues start thinking, how can we do that? How can we put these things in perspective? And one of the approaches that people thought about is the use of allometric scaling. But when you use allometric scaling, you need to keep in mind certain things that limit the utility of this approach. We need to remember that it is empirical. We need to remind each other that there are major differences in the structure, expression, as well as functional activities of various hepatic enzymes and transporters. Of course, we all in the pharmaceutical industry have a commitment towards the 3 R's, which stands for refine, reuse, and reduce. Now, in addition to all of that, allometric scaling may give you a reasonable prediction, maybe accurate prediction of the clearance, as well as other relevant PK parameters. But with that being said, it is not going to define and determine the cause of your high clearance if it is there. You're still going to be in the dark. And because of that, we need to change. We need to find viable human reagents that can enable us to understand this important interplay that we are seeing more and more of it coming in. Because of that, we start thinking about the use of suspension hepatocytes, plated hepatocytes. These are viable too, don't get me wrong. They have reasonable expressions of relevant uptake transporters such as OATP, NTCP, but with that being said, they are not a polarized system. What do we mean by that? These two, they don't express the MRP2 and the PCRP. As a matter of fact, these efflux transporters that are found in the canaliculi, they're actually internalized. So as a result, you are not able to understand the interplay of all of these parameters that we mentioned earlier. So that's why we need to find a more viable tool. This viable tool that we identified as an investment that we need to make is a sandwich culture of human hepatocytes. This tool was established by University of North Carolina, led by our Professor Kim Brouwer. She collaborated very closely with Pfizer, and based on that, we established this tool. The beauty about this tool is the fact that it has a polarized system. Let's walk you through it. As you can see on the surface, there is an expression of OATP transporters, as well as NTCP, as well as other efflux transporters that are found on the sinusoidal membrane. In addition, there is also expression of efflux transporters that are lining the canaliculi. So because of that, we are calling this system a polarized system. In addition, through the use of certain reagents such as the presence and absence of calcium, presence and absence of rifamycin, you are able to deconvolute the interplay of all of these outcomes, such as the biliary clearance, the uptake, as well as the passive, and based on that, you're going to be able to use these inputs to make something meaningful, which is the prediction of the clearance, as we are going to see in a minute. So now, when you look at sandwich culture of human hepatocytes, there has been a major investment in this tool by Pfizer, by the academia, as well as other leading pharmaceutical institutions. The part of the investment was characterizing various enzymes and transporters expression in the sandwich culture. Now we know that there is an expression of the OATP 1B1, 1B3, and 2B1. We know that NTCP expression is comparable to human liver. We also have an understanding of the expressions of CYP enzymes, UGTs, as well as the canalicular efflux transporters. That makes us feel comfortable that when we are going to be basically using the inputs that comes from this important tool, we're going to be able to put it in perspective because we understand what is there. Now, Pfizer leadership is committed to invigorating and the use of mechanistic understanding to enable accurate predictions of the clearance. Part of our deliverables is invigoration and the utility of the PBPK models. So what we have done is part of the investment was developing our own in-house tool where actually we were able to establish the PBPK models where all the organs were perfusion limited and only the liver was permeability limited. We used the inputs of the sandwich culture along with other approaches to build a middle-out approach that will enable an accurate prediction in the drug dispositions and potential interactions. Now, let me show you how we have done that. In Pfizer, before we make any investment, we always stage the investment. We first establish validation that will convince us that it is sound and it works right. So as a result, we started with a sandwich culture where we use the sandwich culture to provide certain inputs, and these inputs are shown over here, which are the uptake clearance, biliary, metabolism, and passive clearance. I need here to emphasize that the metabolism clearance, we got it from human liver microsome and human hepatocytes. This actually, what we have done is we took a list of 12 compounds, and basically we use brute force grid approach to enable the identification of global scaling factor that will enable an accurate predictions of compounds that are eliminated through the same route. As you can see here, when you look at the quality of our prediction, as you will note, is that 90% of the compounds that we predict, their clearance actually is within 2.4-fold. So now we feel comfortable that we have a viable approach. That evaluation of this tool and its effectiveness happens when we actually use a compound and we prospectively predicted the clearance of the molecule. To our happiness and excitement, actually our clearance, I can comfortably say that it was spot on. As you can see, it is right on the line of unity, which actually makes us excited because, interestingly, if we ended up using monkey single species scaling, this compound was predicting that its clearance was going to be hepatic blood flow limited, and as a result, we may ended up actually killing the compound. The utility of sandwich culture human hepatocytes and integrating it into a viable, PBPK validated model enables us to keep this compound and to move forward. And to further expand on this business, we actually were able to show that our KBUU prediction using PET imaging was consistent with the outcome of our scholarship, which made us very happy and excited about the progress that we made thus far. This compound that I'm talking about was in advanced lead. It was going into human, and that's basically what we have done. We, inside our discovery team, had a different question. Our colleagues in medicinal chemistry, they start asking an important question, which is, what are the parameters that we need to change to have a compound that is appropriate for BID dosing at least, and also have nice hepatoselectivity? They start asking, provide us with guidance. What are the changes that you are supposed to be making, and what is the value? So now what we have done is we have done a sensitivity analysis. We had a compound where we know what's basically the prediction based on the input that we obtained from sandwich culture. And we start saying, okay, if we change only the uptake clearance and maintain everything the same, how this changes in the uptake clearance is going to affect the half-life. As you can see here, as you are increasing uptake clearance, this is the clearance that scales from the sandwich culture, you're actually going to observe that there is a significant reduction in the half-life. With the same token, increasing the uptake clearance will end up actually increasing KBUU. As you can see, there are two things that are actually working against each other. So as a result, we need to find a balance between the two. And that's where you need to start thinking about the refinement. What we have done is we expanded this sensitivity analysis by basically assessing the impact of other parameters, and based on that, we came up with this simple cartoon. This cartoon is suggesting to you, if you would like to refine dose, you need to be mindful of the clearance since it's going to affect your half-life, and you need to be mindful of the KBUU, especially if your target is the liver. Now, what's interesting that these changes in any of these parameters most of the time is a sword with two edges. One of them can help you and the other can hurt you. So, for example, if you want to lower your clearance, you need to lower your uptake. But at the same token, if you're going to be lowering uptake, that's actually going to end up lowering your KBUU. So you need to find the right balance, and that happens by understanding and comprehending the interplay of all of these parameters. So now, in addition to all of that, we expanded this investment by also seeing how we can predict the impact of disease states on the clearance of the molecules, and also to prospectively provide an approach to predict the impact of genetic variations and 1v1 on the exposure of these molecules. You have the two references for two articles for your reference if you're interested in following up on them. So that was our investment from an in-house established model. Now let's talk about how we have used Simcyp in enabling the prediction of transporter-mediated clearance drug-drug interaction. I like, when I'm going to be approaching this ECCS class 1B or 3B, which is basically the compounds that have active hepatic uptake as a rate determining step, I like to have this cartoon. It will give you an idea about where you stand and how you put these things in perspective. As you can see here, we started with Repaglinide. As I said earlier, we typically first validate the concept before we make the investments of prospective prediction. So we use Repaglinide as an anchor of our thinking. As you can see, Repaglinide, which is a class 1B compound, when it's going to be taken up into the liver, it's going to happen by an active uptake mechanism that's mediated by 1B1 and 1B3. Once it's inside the liver, it's going to break down and metabolize by CYP2C8 and to a lesser extent by 3A4. The compound has a complete absorption and minimal renal elimination. In fact, we have actually established the ability and the utilization of Simcyp to make a prediction of the elimination of Repaglinide. And as you can see here, we were able to capture nicely the outcome of the reported data. The green triangle is the PBPK predicted outcome, and the red circles are what's observed. As you can see, we were able to replicate what we have observed. And as a result, we were very comfortable now to use Simcyp because we established a roadmap of putting things in perspective. Now, how did we use that? We actually have a compound that we have identified as a class 3B. This compound gets inside the liver by active uptake mediated by 1B1 and 1B3. And once it's inside the liver, it's going to be eliminated in the bile as unchanged drug through PCRP. We know that this compound has some renal elimination and its absorption is optimal. So now, the very first thing, and based on the learnings of Repaglinide as well as other compounds that we have investigated, we found out that the sandwich culture inputs that we are getting, we need to multiply by a scaling factor of 2, similar to what we have, similar to the trend that we have observed with Repaglinide, and we were able to capture the outcome of various doses. Now, the good story is, based on that, we were asking ourselves, what's going to be the changes in the plasma exposure of our compounds in the presence of an OETB inhibitor such as cyclosporine? We prospectively predicted that the change is going to be 3.1-fold increase in the AUC. Interestingly, it was very similar and comparable to the observed AUC change of 2.74. So the good story about that, the investment that we made in putting things in perspective and learning from compounds with an established track record, it enabled us to accurately, prospectively predicting the elimination and the drug interactions of our compounds, which was highly appreciated by our clin pharm as well as other members of the development team. Now, what we are calling for. I am calling for a change in how we are approaching our drug disposition. We should not have one size fit all where all the compounds are going to be evaluated using the same approach. The assessment and the evaluation should be driven by the rate determining step if we are trying to understand the systemic exposure of the molecules. This is where extended clearance classification system, the ECCS, will come in. It will enable you to understand what is the rate determining step of your compound evaluation. And based on that, you're going to be defining what kind of investments and what are the tools that will enable you to establish a QSAR to weed out compounds with issues around its clearance. Once we have identified the compounds, we're going to be able to generate inputs such as what we have done with the sandwich culture human hepatocyte. These inputs will be feeding into a PBPK model, which will enable us with biological inputs to define the dose and to make a prediction of what is the potential drug-drug interactions of our compounds what's the potential for food-drug interactions, not to mention impact of pharmacogenomics and disease state on the compound exposure and efficacy. Now, the concluding remarks. As we all can see, there is a major shift in our understanding of drug disposition that require us to change how we are running the business of the clearance screening studies. Sandwich culture was clearly shown to identify and determine the various inputs of the extended clearance equations, which based on that, we were able to validate and to use prospectively to make the predictions for relevant compounds of interest. Of course, integrating sandwich culture in modeling and simulation tools is very powerful in enabling an implementation of bottom-up and top-down approaches to successfully predict the clearance of the drug interactions with active hepatic uptake, which is ECCS class 1B and 3B. I would like to end my presentations by a very important slide, which is acknowledgments. I would like to thank all my team members in providing a viable environment that fosters open discussions and debate and will continue to improve how we are approaching our business. With that, I would like to say thank you for this opportunity and thank you for listening.
Thank you, Ayman, for this excellent presentation, a very comprehensive discussion. I would just like to remind the audience that if you have questions regarding Ayman's presentation, to please email those while I am speaking. I would like to continue on with a discussion of some key considerations in selection of a predictive hepatic model, and specifically for drug discovery and development. And there are a couple of issues that Ayman briefly mentioned that I want to expand upon in this discussion, particularly the focus on the basolateral efflux transporters and their role in hepatobiliary drug disposition, bile acid disposition, and disease state-associated alterations in these basolateral transporters. And then secondly, to focus on the regulatory pathways that are being discussed that are very important to these transporters, and then key considerations in model selection. So I think everybody's familiar with the physiology of the hepatocyte, the localization of these uptake transporters in blue, and as Ayman has discussed, the canalicular efflux transporters in purple here that mediate the biliary excretion of both drugs as well as metabolites and, importantly, endogenous compounds. But less well studied are the basolateral efflux transporters, which clearly, as we are learning, play a very important role in the hepatic disposition of drugs as well as endogenous compounds, specifically MRBs 3 and 4 and OST alpha beta for bile acid efflux. Now, I think we first of all really became quite aware of the importance of the basolateral efflux transporters with the nice, elegant work that was published by Schinkel's group, depicted here in this slide, where we see the hepatocyte hopping of the bilirubin glucuronide. So unconjugated bilirubin is taken up by the OATPs into the hepatocyte, metabolized to the glucuronide conjugate, and excreted into bile. Now, a portion of the bilirubin glucuronide that is not excreted into bile is fluxed back out into the portal circulation via the MRP3, and then is taken back up by OATP 1B1, 1B3 into the hepatocyte for a subsequent opportunity for biliary excretion, and again, residual that isn't excreted into bile is transported back out, and so forth. And this allows for a very elegant, very efficient system for hepatic detoxification of the bilirubin glucuronide. Now, we were curious about the role of these basolateral efflux transporters in this hepatocyte hopping for drug disposition, and so we specifically focused on rosuvastatin, which we know is a good probe substrate. It's primarily eliminated by the liver. It undergoes minimal metabolism and low passive permeability, so it's a transporter that dominates. And you can see here the systems that are predominantly responsible for the uptake of rosuvastatin, primarily OATP MRPs, and excretion into bile via MRP2 and BCRP. And the contribution of the basolateral efflux transporters, specifically MRP3 and 4, was not known at the time that we conducted this work. So we use the sandwich cultured hepatocyte system, the B-Clear technology, and a standard incubation with or without calcium, an uptake phase depicted here in purple, and then also a efflux, using a specialized efflux protocol where we could measure in the standard calcium-containing buffer or the calcium-free buffer from the cellular compartment as depicted in blue, and also from the medium compartment in the dashed blue line. And then using this approach coupled with pharmacokinetic modeling, we were able to recover the clearance pathways for the uptake, the basolateral efflux, as well as the biliary clearance for rosuvastatin. And the results of the experimental protocol and the modeling revealed some very interesting data in the sandwich cultured human hepatocytes. You can see here the relative contribution of basolateral efflux to overall hepatic clearance versus biliary clearance. And the biliary and basolateral clearance values are shown here in the table. So again, about 70-some% of the rosuvastatin taken up into the hepatocyte is fluxed back out by the basolateral efflux transporters into the medium, and a much smaller percentage going into bile. And in the rat, sandwich cultured hepatocytes, we saw a more even distribution of the biliary and basolateral clearance here, the values being quite similar, so about a 50-50 movement of the rosuvastatin into bile versus back into the medium. So I think this is a nice example that emphasizes the importance of the basolateral efflux transporters in hepatic disposition of rosuvastatin and the use of the sandwich cultured hepatocyte B-Clear technology to quantify these various clearance pathways. And subsequent work in our laboratory revealed that rosuvastatin is a substrate for MRP4, which likely mediates, at least to some extent, the basolateral efflux in human liver. Now, I'd like to just pause for a minute and emphasize the importance of being able to quantify the biliary excretion versus the basolateral efflux, which one can do using the B-Clear technology, but is not possible when one uses a single well methodology. And let me convince you of this with some data. So first of all, as you know, the drugs such as either a parent or metabolite that might have a significant component of basolateral excretion, like rosuvastatin, relative to biliary excretion, if one was just measuring what was coming out into the medium, you would expect there to be a significant overestimate if you were considering that as your biliary clearance. I think a nice example is with naloxone glucuronide. It's rapidly taken up and metabolized by the hepatocyte to a glucuronide conjugate, and we know that in humans, naloxone glucuronide is primarily eliminated in the urine. The single well methodology, as depicted here in red, would predict that the biliary excretion, if you will, by just measuring what's in the medium, would be quite high, when in fact the biliary excretion of naloxone glucuronide in vivo and in the B-Clear system is very low. And we also have generated similar data with morphine glucuronide, and I'll show you some clinical data to further support that in a few minutes. So another important issue is that the single well methodologies are not able to identify circulating metabolites that would be excreted back into the blood via the basolateral efflux transporters, as opposed to the ability to assess that in the sandwich cultured hepatocyte system with the B-Clear technology. And it's not just rosuvastatin, but other statins, actually I think many other substrates, including bile acids, where basolateral efflux plays an important role in hepatic disposition. Now, in addition to drug disposition, we know that the basolateral efflux process is important for endogenous compounds, as we showed in the beginning with bilirubin glucuronide, and also with bile acids. For example, bile acids are taken up into the hepatocyte by sodium tartrate co-transporting polypeptide. They are predominantly excreted in human hepatocytes into bile by the bile salt export pump, as depicted in this schematic. And we know that some drugs, for example, triglyceridone and the sulfate conjugate of triglyceridone, inhibit the bile salt export pump. And this results in accumulation of bile acids within the hepatocyte. Now, interestingly, triglyceridone sulfate is a very potent inhibitor, as is triglyceridone of the basolateral efflux transporters, as depicted here, and we have data to demonstrate that this is one mechanism of triglyceridone-mediated hepatotoxicity. So again, the importance of the basolateral efflux transporters in bile acid accumulation in the hepatocyte as a mechanism of drug-induced liver injury, I think, cannot be overemphasized. So it's important to think about the transporter-mediated bile acid-drug interactions that may be occurring, and to do so, one needs to think about the relative contribution of the biliary versus basolateral efflux pathways for bile acids, and what impact inhibition of one or both of these processes might have on the accumulation of bile acids, for example, within the hepatocyte. Using a similar study design as described with rosuvastatin, we looked at the effect of basolateral efflux and biliary excretion on the contribution to hepatic tarcholate accumulation, again in the sandwich cultured human hepatocytes first. The data, as shown here, reveals much what we would expect, that the majority of the tarcholate is excreted into bile via BSEP, about 80% approximately undergoing biliary excretion, and some modest basolateral efflux. In contrast, with rat sandwich cultured hepatocytes, we see a greater contribution of the basolateral efflux pathway to overall tarcholate disposition in the sandwich cultured rat hepatocytes. And you can see here the biliary and basolateral clearance values are more similar than in the case of the human sandwich cultured hepatocytes. Now, the reason this is important is if you think about a compound that might be inhibiting BSEP, for example, and how one might be making predictions of the impact of bile acid accumulation within the hepatocyte. In rat sandwich cultured hepatocytes, inhibition of BSEP will only have a modest effect on hepatic bile acid accumulation within the cell, as depicted here. A tenfold impairment in the biliary clearance of tarcholate causes a very modest increase in intracellular concentrations of the tarcholate. But in contrast, because the biliary excretion process plays so much greater role in human sandwich cultured hepatocytes, inhibition of this pathway would result in a much more significant increase in the cellular accumulation of the tarcholate. And of course, we know that bile acids are important regulators of the nuclear receptor, and there are many other changes that may be occurring to modulate both the uptake as well as the excretion and the synthesis of bile acids, and I'll move on to talk about that in a few minutes. But I think that's another example of the importance of basolateral efflux transporters. We know that many disease states may influence the expression of these basolateral efflux transporters. For example, in obstructive cholestasis, MRP3 protein expression is upregulated. Also in HBV-related cirrhosis, we see induction of MRP4. And even in non-alcoholic steatohepatitis, some very nice data from the Charrington lab, Rhiannon and Hardwick, showed clearly in patients with NASH an impressive induction in both MRP3 and MRP4. And clinical data to support the impact of this on drug disposition was generated by our laboratory looking at morphine disposition. IV administration of morphine, we see no change in the concentration time profile for morphine itself in healthy subjects in blue versus NASH subjects in red. And the no difference in Cmax and AUC as depicted there. But in the generated morphine glucuronide concentration time profile, both for the morphine 3 and morphine 6 glucuronide, serum concentrations as depicted in red for the NASH patients are significantly higher in the kinetic parameters shown below. We know, as mentioned, that bile acids are regulators of the nuclear receptors. Intracellular accumulation of bile acids will increase BSEP expression, but if there's some impairment in BSEP expression, these other basolateral efflux transporters, including OST alpha beta, will be significantly increased, and this is depicted nicely in the gene expression changes observed in human sandwich cultured hepatocytes following exposure to chenodeoxycholic acid for 72 hours. You can see the increased BSEP expression, again by mRNA here, and also OST alpha and beta, very significant increases with the exposure to the chenodeoxycholic acid. And data is not shown here, but CYP7A1 mediated bile acid synthesis is correspondingly decreased with CDCA 72-hour exposure. The net effect of the sandwich cultured hepatocyte system following bile acid exposure, at least with CDCA, is to decrease the intracellular concentrations of the endogenous bile acid. So when we think about a predictive in vivo relevant hepatic model system, there are a number of components that need to be present and functioning in a physiologically relevant manner. Of course, we need to have the uptake transporters properly localized along with the biliary and basolateral efflux transporters. And certainly there is some debate in the literature about the suspended hepatocytes versus sandwich cultured hepatocytes. I think that if you look carefully at the literature, suspended hepatocytes, following isolation of the hepatocytes, the canalicular proteins are internalized. Now, some published data shows significant remnant clumps of hepatocytes, doublets or triplets, where there are these residual canalicular compartments with some functioning transporters, presumably. But in properly isolated hepatocytes, where you don't have a lot of these clumps or doublets or triplets, the canalicular proteins are internalized and they are not on the membrane to be functional. And one needs to have a polarized system with those proteins properly localized to accurately quantify the biliary excretion and to distinguish that from the basolateral efflux transporters, which we've seen can contribute significantly to drug as well as bile acid disposition. One also needs a metabolically functional system, and also necessary is the ability to respond to the regulatory responses that one would see with bile acid exposure or other drugs which may be causing regulatory pathways to be altered. And again, this is an area where I think further research is needed. And with the proper system, one can accurately measure intracellular concentrations of the species of interest. So to summarize, basolateral efflux transporters play a key role in the hepatic and systemic exposure of drugs and endogenous compounds and metabolites. The contribution of biliary and basolateral excretion to overall hepatic efflux is both compound and species dependent, and is dependent also on the model system that you choose to use. One needs to consider the effects of drug interactions and disease state alterations on these transporters, in particular the basolateral efflux transporters. There can be inhibition as well as induction these pathways. And finally, it's clear that the sandwich cultured hepatocyte system does represent a physiologically relevant system that integrates these key components necessary for accurately predicting hepatic clearance. And when one couples the data with modeling and simulation, it's a very powerful tool to assess the processes that are involved in hepatic drug disposition, drug interactions, and drug-induced liver injury. And with that, I would like to thank my current and former students and postdoctoral fellows. Those highlighted in blue are the ones whose data I emphasize today, some of my faculty colleagues for the contributions to this work, to also mention my conflict of interest, and also acknowledge the funding from NIH as well as other sources that made this work possible. And with that, I'd be happy to open this up for questions. Thank you.
Thank you, Kim. And to all of you on the line, please continue to submit your questions. We have a question here from Patrice. Ayman, I think I'll give this one to you. Concerning the global scaling factors, mean values were presented. Are these consistent across compounds and can be used, I've lost it, used readily? Or is some understanding of specific molecule required to get the correct scaling factor?
So when we
take that one?
Yeah, absolutely. First, thank you, Patricia, for this good question. The short answer is these global scaling factors are going to be used prospectively for compounds that fit the criteria, which is the fact that they have active uptake mediated by OATP as a rate determining step. They are not going to be compound specific, and that's why we are able to have a prospective prediction. This is a major improvement that we have done relative to our first paper that we have published in 2012, where it was compound specific. Short answer, yes, it is used for compounds as long as they fit the criteria of having active hepatic uptake as a rate determining, and it's not compound specific.
Thank you, Ayman.
Thank you.
Kim, a question for you. Some articles do report presence of efflux transporters in suspension as well, and sometimes confuses researchers. What do you think about that?
So I see that Dr. Chatterjee asked that question, and as I had mentioned rather hurriedly in the presentation, these canalicular transport proteins clearly internalize when one isolates the hepatocytes. And Dan Bowe had published work previously showing the intracellular localization of these transport proteins. It's well known from Wynne Arias's work and other investigators that the transporters, the biliary transporters, so BSEP in particular, or P-glycoprotein, they reside in subapical compartments in vesicle-like structures, and they can be very rapidly moved on or off the membrane by signaling pathways in the hepatocyte. So, for example, cyclic AMP-mediated pathways that are stimulated by bile acids within minutes can move the transporter to the membrane surface. But when those proteins are not on the surface, they are not functional. So the data, again, I would refer the audience to some of Dr. Arias's work. The data clearly show that the proteins have to be, that the hepatocyte has to be polarized, and in order for the proteins to be on the membrane, for those to be functional and to have bile flow and pumping of substrates into the bile compartment. Now, you may see movement of molecules that you think would be excreted into bile into the medium. For example, I'll pick on morphine glucuronide, and you saw from the clinical data morphine glucuronide appears in the serum in patients. And that happens by MRP3, which moves the glucuronide conjugate across the basolateral membrane into the serum. So likewise, when you're incubating suspended hepatocytes and add morphine and you see morphine glucuronide in the medium, that's getting there via MRP3, which does not move off the membrane when the hepatocytes are isolated. But the isolation process with collagenase digestion basically rips apart the canalicular domains and causes those canalicular proteins to internalize.
Thank you, Kim. I think we have time for just one more question from Vineet. Which system is better for drug disposition prediction, hepatocyte sandwich culture or hepatocyte co-culture? Big question there. Kim, do you want to take that one?
Ayman, were you going to say something?
Yeah, if you want, I can take it.
Yeah, please do.
So in my mind, when we are going to say which method is appropriate, we need first to ask ourselves did we characterize and validate the method before we make the investment? So when I'm looking at the sandwich culture of human hepatocytes, I think we have done our extensive due diligence, and we have a track record that we understand the basis and all the expressions of and activities of various enzymes and transporters that are available in this system. And also we have used extensive database to enable a prediction of the compound elimination and clearance. I know that this is very well established for the sandwich culture. Now, for the co-culture, till that is going to be established, till we are going to have a good understanding of the transporter expressions, and also we will do the necessary validation and we will put it in a viable PBPK models where we can prospectively predict. Till that's going to happen, sandwich culture is what is standing at this point, in my opinion.
Thank you, Ayman. This brings us to the end of our hour, and I really want to thank you, Ayman and Kim, for spending time with us today. Really appreciate your insights and the information you shared. And a big thank you also to everyone who has joined us. We'll follow up with each of you and send you a copy of the slides, and hope to continue to discuss with you the subject area and how we can partner in the future. Thank you all.